Running River is home to an endangered, soon-to-be classified species of rainbow fish. The river flows from the top of the Paluma Range and connects to the Bodied River around 46 kilometres to the west. This video aims to bring you some insight into the project and update you on just some of the work they've been doing.
So the Running River Rainbowfish story really started with Ray Leggett, who was a member of the Australian New Guinea Fishers Association. He was up here doing a survey of the Bodied River with the Queensland Museum, and they were the first to collect in Running River that we know of, and he recognised that the Running River rainbowfish and some of the other rainbowfish populations in the Bodied River were different to other rainbowfish. And it's never really quite stuck in scientific circles, so it's always remained fairly poorly explored. But the aquarium hobby, with all the native fish enthusiasts, have always kind of had a thing for the Running River rainbowfish because it's such a different looking fish. And so that's always kind of piqued our interest from a more scientific perspective, that we really need to sort out what's going on with these various populations up here as to, are they really different species or are they just variants? And so myself, Peter Unmack and Dr. Michael Hammer from the Northern Territory Museum did an expedition up here looking at rainbow fishes and other fishes, and we came through Running River to collect some samples. We noticed when we were catching the fish that there were a few other individuals that looked like regular eastern rainbow fish, which is otherwise found in other parts of the Bodied River system and up and down the east coast of Australia. We're kind of like, well, okay, there's a couple in there. We didn't really think too much of it because we're not intimately familiar with the river and the fish. But we did know there was supposed to be no rainbow fish found upstream. So Running River rainbow fish is only found between two gorges on Running River, and that restricts other rainbow fish from coming up from downstream, and it stops them getting further upstream. So we knew there was supposed to be no native rainbow fish above the upper gorge on Running River. And then when we popped up above to sample some other fishes, the first fish we caught were rainbow fish, and we're like, uh-oh, this is bad. Now we know where these strange looking fish we were catching downstream were coming from.
I was informed by Peter Unmack about the presence of an introduced eastern rainbow fish into an area previously devoid of any sort of rainbow fish within the Running River, and he contacted me because of my history monitoring freshwater fish within the Running River, and wanted to know some of the background and dates as far back as I could remember.
At that point we realized the Running River rainbow fish is probably on the verge of extinction, because anytime you mix two rainbow fish species, they will usually hybridize together. So if somebody moves some from somewhere else into a new population, they will mix with whatever rainbow fish is there, more than likely. And that's exactly what has happened here.
And it's been really fortunate in that because we've had some pretty awesome knowledge within the project team in terms of the genetics and taxonomy and the ecology of these beasties, that we've been able to put this project together so quickly.
So one of the quirky things about rainbow fishes is they tend to really differentiate between creeks. So you'll get all these different slight colour variants that arise in different localities. Even though they're all the same species, you can often look at them and say, oh, I know that's from the Proserpine River, or you might say, oh, I know this is from the Mulgrave River, or, so that they do evolve slight differences between them in colour and fin shape and size that allow you to distinguish them. And that was one of the problems with Running River rainbow fish. Is it just a colour variant that's a little bit different, or is it actually a different species? And that's often a difficult boundary to work out, but that's one of the key things we were focused on.
Peter Unmack from the University of Canberra invited me along to come on to survey sections of the Running River and associated tributaries to try and find basically confirm the status of or presence of the Running River rainbow and in any of the tributaries that run into the Running River, and invited me along to come out into the field with him and a few other colleagues in late 2015. So we went along and surveyed a few creeks above barriers to look for suitable translocation sites, as well as to collect brood stock from the Running River for Peter to take back to the University of Canberra in order to get tested to see if they're, well, to make sure that they were pure stock for inclusion into the breeding program down at the University of Canberra.
So once we worked out that eastern rainbow fish had been introduced upstream and had moved down into Running River, we decided to remove a little bit over 50 fish to bring back to Canberra as brood stock. And the first key thing we did was set them up so that we could genotype them. So we had to make sure these are pure Running River rainbow fish and they're not hybrids, because you don't want to try and save a species. You know, you want to make sure you've got the right stock to start with. So we genotyped all the fish, and then we were able to use that information to determine how to pair up the different fish. So our collaborators at Flinders University did a series of analyses to tell us which fish to mix with which fish to maximise genetic diversity. And so that was a really key novel part that's rarely incorporated into these sort of programs. And so then from there we just started breeding the fish, and the goal was to try and produce an equal number of babies from each parent fish. So we had an even mix of the genetics across the parents and the offspring. So when we put them out in the wild, we've got as best representation of those 50 fish as possible. We needed to find other partners, so one of the key problems was we could breed a lot of fish at Canberra, but we didn't have the facilities to raise fish. So I contacted my collaborators at James Cook University, and luckily enough they have a nice aquaculture facility that they were able to set up some ponds in and maintain some fish. So we started shipping up babies, and then with the help of Jason Schaefer and Damien Burrows, they got going at Freshwater at James Cook. They got going with raising up these fish.
They've managed to breed up quite a number of fish down there, which they have subsequently sent back up to us here at JCU, Freshwater, at our grow out facilities here. At the moment we have close to 3,000 about 2 centimeter long Running River rainbows that we're growing out for that purpose.
And then that enabled Carl, once he got going up here, the fish are already here ready waiting for him to take them out to the creek. And so we were really, really lucky that Carl came along at the right time in this project.
So I first got involved with with the Running River Rainbowfish project in 2016. I met Peter Unmack, my current supervisor and the head of the project, at an Ank for Queensland meeting. Peter was giving a talk about the Running River Rainbowfish project. Afterwards, Peter and I got chatting, and eventually led to me getting involved in the project as well.
And Carl came up to me after the meeting and said, "Oh, I'm really interested in this. I'm kind of interested in pursuing further study on this. You know, there's some opportunities." And I was like, "Absolutely." Carl's turned out to be really good. It's not easy working up here, and so basically he got sent up here in this remote area with relatively little outside contact, and his job was to go survey the creek, go find some pools we can release fish in and do a little bit of science. So he tracked around for a few weeks checking out the pools, hiking up and down the creek, all in November, December in hot weather and difficult conditions. And
one of the biggest difficulties in finding these habitats was the terrain itself. So it takes about 40 minutes to get into the creek. And that's just up and down exposed bedrock, muddy sections of track. You need a four-wheel drive to get in there, sort of thing. And then once you get in, it takes probably another 50 minutes to get from one end of the track to the other. Once you are in there and you're walking around in the creek, it's mostly exposed, smoothed granite bedrock, which means with the slightest bit of rain, it gets quite slippery. In addition to that, it's very hilly, canyony, rocky country, so lots of waterfalls, lots of sheer cliffs, lots of cobble fields, so you got to watch your step while moving through it. Eventually I found 10 pools that I thought fit the requirements for my release sites. Deception Creek and Puzzle Creek are both primarily on land owned by the Australian Wildlife Conservancy, which is a brilliant non-for-profit that aims to protect and conserve and in many places restore Australia's native biodiversity and keep it there for future generations to hopefully enjoy.
There's been a little bit of work with translocations in some Australian native fishes, but it's not a supremely common thing outside of angling species. So fish like trout cod or Murray cod or Mary River cod, Macquarie perch, have all been stocked widely, but they're all big angling species, so they have a whole group of people interested in them that the little native fishes don't have. So there's been a bit of work done on several galaxiids that have been threatened from various causes that have been moved to new locations. But this is a first for rainbow fish, essentially, where we're actually putting them into new places where they didn't previously exist as part of their conservation actions.
In the search for new habitat for running river rainbowfish, it's fortunate that Mount Zero Taravale is right next door, so the boundary is next to one of the sections of gorge where running river rainbowfish originally lived, and there are a couple of tributary creeks that run into Running River, and the upper habitat of these creeks has been isolated by waterfalls, essentially, that are on the edge of the where the range falls down towards the Bodied River, and those creeks have got habitat where we think that the rainbow fish will be able to live and thrive. So there are no rainbow fish currently above those waterfalls, so they make perfect translocation sites.
Again, there's actually many elements of this project that came together very fortuitously, and if any of them had have failed, the project would have been far more difficult to pull off.
And we're very lucky in that we've had a landholder that had the right sort of creeks available within the catchment that the fish originally came from.
One of the biggest benefits of Deception and Puzzle Creek is that they flow into Running River. They don't flow into the 13-kilometre stretch that the Running River Rainbowfish was initially natively found in, but Puzzle Creek flows in just above, and Deception Creek flows in just below the 13-kilometre stretch that it was originally confined to. The best part about that is when you introduce fish into an upstream area, they will inevitably end up moving downstream. So if they're just moving downstream into where they were before, there's no negative consequences of that. Whereas if we would put them into a creek, on the other side of the Bodied River or in a different river system, eventually they'd end up moving downstream and causing the same problem that the introduced population of eastern rainbow fish has done to the Running River rainbowfish.
So one of the key things we're trying to work out here is how do you do a reintroduction of rainbow fish or a translocation of rainbow fish? It's not, you don't just pick up a book and it tells you release this many fish at this time of year and this size fish, and so we're kind of working in the dark and our best guesses.
There is not a lot of information regarding the factors influencing the success of introductions of small-bodied freshwater fish in Australia or across the world. There's been a bit regarding larger longer-lived fish species such as trout and cod, but not a lot regarding smaller-bodied fish. Of course, they don't get as much attention because they're not an angling species. They're not big or impressive, and people don't eat them. So one of the biggest problems is these small-bodied things, like rainbow fish, hardyheads, they only live for 1 or 2 years. They hatch, they grow up, they breed, they die in the span of 1 or 2 years. Some studies on trout releases have shown that upwards of 90% of fish released die within the first week. That means someone says they released 100,000 fish into the Cotter Dam or the Murray River or wherever. 90,000 of those fish have died within the first week. The reason for that is hatchery reared fish grow up in a featureless pool, featureless pond, crammed in as many as they can, and then they get fed pellets that float on the surface. When they go into the wild, they are not the only fish in a featureless pond. They are one of many species of fish in a pond with different deep sections, shallow sections, vegetated sections, shady sections, open water sections, and there is no one throwing in ready to eat food for them. They have to go out, they have to find that food.
We've taken quite a few steps. So there's predators that live in these creeks, such as spangled perch and purple spotted gudgeons and things. And so we've tried to take steps in how we raise the fish so that they get some degree of understanding that there are predators out there and they can hurt them. So we've done some predator training with the fish in the tubs. We've provided them with rocks and other cover so they have escape mechanisms. We've tried to feed them natural food so they recognise what that is instead of just artificial foods. And these things all potentially contribute to increasing survival.
Fish living in a stream are always swimming against that stream current. Fish living in an indoor hatchery pond are not swimming against any current. There's no threat of them getting washed away or anything like that. They just sit in their pond and wait for someone to throw some food in. And as you can imagine, getting put into the real world is a really big shock. For most, it's too much. So what I was doing is trying to figure out some of the best ways that you can prepare small-bodied fish for life in the real world, as it might be called. One of the ways we did that was trying to teach them about predation events, or predators, teach them about predators. To do that, we took a spangled perch, and for 15 minutes a day, 7 days prior to release, we would put a spangled perch in a mesh box about that big, and we would place him in their rearing pond. So the box was made so that the rainbow fish could swim in and out, but the spangled perch was stuck within it. And as we put him in, or her in, we also put in an ice block made of what we call rainbow fish essence. So there's been some research done that shows when social fish that live in groups, when they are attacked and the skin is broken, it releases an alarm pheromone which alerts all their conspecifics or all the other members of their species that there's something bad going on and that they need to be afraid or on their guard. So rainbow fish essence, as it's called, we euthanize one rainbow fish, then we crush it up in some water, strain off the rainbow fish and leave the rest of the water, and we freeze that, and then we have an ice cube of rainbow fish essence. So the idea is the fish learn to associate the stimulus of the sight, the smell of a spangled perch with the smell of predation of this alarm pheromone, so that they know to associate the spangled perch with a predation event, with something that they should be worried about. So that when we release them into the wild, they don't just all swim out in the open and just get eaten by spangled perch.
So those releases went really well. The fish were able to get in there, get some spawns off, get some fry going, and then get some rainfall events so they could disperse. And then Carl's follow-up surveys on this trip have been fantastic. You walk up to the pools and you just look at the pool and you're like, oh, there's a rainbow fish, there's a rainbow fish, there's a rainbow fish, oh, there's 20, oh, there's a bunch of fry. So the fish have bred up and done really well.
It's important to monitor after release. So it's all well and good to say you released a whole bunch of fish into the creek, but it's actually worthless if you don't actually know whether they've survived or not. So all good conservation programs have a monitoring component afterwards to know that you've actually done something and actually achieved what you set out to achieve. So what I did was a snorkel survey where I would snorkel down the left-hand side of the pool, and back down the right-hand side of the pool, and then back down the middle. I'd cover as much of it as possible, where I would look for the fish that I'd release, and I'd count the number that I saw. I'd count out the total number that I saw in general, and also count the total number that I saw at any one point. And that allows me to make a relative measure of abundance. So I can't tell you exactly how many are in there, but I can tell you that there's more in this pool than there are in that pool. So when fish were first released into a pool, they're frightened, they're scared, they're timid, they're not confident within the environment, so they would often stick to one point. But after about 2 weeks, fish would start moving around, and they would no longer be getting no longer be getting constantly chased by predators such as spangled perch and gudgeons and turtles and things like that, and would actually be nonchalantly swimming around with their former predator. I'd have to be swimming around at a constant pace to stop the fish that I'd already passed from swimming in front of me again. So in the first few weeks, most of the fish would be hunkered down in one little spot that they just thought was great. That was the best part of the pond. It was often shallow, sandy, and warm. And as I would swim past that point, they would often come out to greet me, as it were. Probably weren't greeting me, they were just checking me out to see if I was a friend, foe, if I had any food on me, you know, fish stuff. And eventually they would start swimming around me in circles. So it was very important that I kept a constant rate of movement, so that I would swim past, count them, and that they wouldn't start swimming around me in circles, because then I would end up recounting fish or not being able to count the number of fish, because I'd start here and it'd just be a constant circle and wouldn't know when to stop again. Once they were in that, once they had that level of confidence to swim around the pool openly and freely, they would then just start following me. So I might start at one end, get to halfway, turn around, and then just be this huge wall of fish swimming around behind me. In the uppermost pool, I released 250 fish per pool. When I would go snorkeling, so you never saw 250 fish in the days after. It was a huge, the pools are over 150 metres long, and 250 sounds like a lot, but the fish are all about that big when they go in. So you're not going to see 250. Even if all 250 were released and they survived no problems, you're not going to see or count 250, because they'll be in amongst the weed. They'll be in spots that you just can't get to as a snorkeler. They'll be all over the place. But on a good day, I would see around 150 fish. When I went back this month, which is probably about 3-4 months later. Yeah. When I went back 4 months later, I was swimming around and I was seeing upwards of 400 fish, counting upwards of 400 fish. And I would be swimming along and I would turn around and I would see schools of 100, 150 fish swimming around behind me. It was an amazing feeling just to see something, to see what was a critically endangered species, or technically now extinct in the wild. It was surreal to see something swimming around and doing well.
One of the problems we have, or we had, is that because we didn't have a formal national government status for this species, you couldn't attract funding from government, and so the funding for this has all come from universities or our in-kind time, or from aquarium societies or whatever.
It's amazing that this project's essentially been supported by fish keepers from all across the world. Rainbow fish are a very popular aquarium fish. Nearly every pet store in the world has them. They're very pretty fish. They're easy to keep in captivity. They're wonderful to watch. And so I approached various fish clubs in North America and Europe and Australia about the project, and many of them were very happy to donate. Quite a lot of individuals donated funds as well. So we ended up raising a little bit over $11,000 so far to help support the project. And that's just kind of unprecedented.
So it was a surreal feeling to see this fish swimming around in the wild and going forth and multiplying and prospering, and knowing that none of that would have been happening if I hadn't spent all those hours and late nights carting buckets from the car to the creek. It was absolutely amazing.
I just sort of like to talk a little bit about some of the uniqueness of this project, in that we've managed to get up here and do a whole lot of work, and it appears that we have a successful outcome in a very short period of time, and that's quite unusual for threatened fish projects in Australia. Threatened fish are largely forgotten. And you know, there's a whole lot of problems in that threatened species declaration process that we've bypassed a little bit with running river rainbowfish. So because it's a newly identified species, it hasn't been formally described yet, and to go through even those steps takes quite an amount of time and effort.
So no one actually ever wants to have to save a species, but it's neat to be in a position where when things go bad, you're in a position where you can actually step up and take some actions and actually have some positive outcomes, so that, you know, my kids and other fish keepers and other fish enthusiasts can come back to these places and still see this fish swimming around in the wild, rather than just looking at dead museum specimens or reading about them in a book. And that's really what it's all about, is, you know, keeping this thing for future generations. You know, it may not excite some people, but there's people out there that get really excited about this sort of fish, and, you know, and that's what drives my passion about what I do. And so for me, it's a really cool thing to be able to contribute something back to the fish world. So the project's not over. We have more work to do, and there's other rainbow fishes that we have active research projects on as well that are in similar dire straits to Running River. And so if people would like to donate, so our crowdfunding site can be found if you Google Running River Rainbowfish, Canberra. It's run through the University of Canberra Foundation. There's a spot there where you can go and donate via PayPal, or through your bank account details, credit card details, and all the funds go directly to the conservation work. There's no 15% gets siphoned off for anything else. 100% goes to the project, which is really good.
If you'd like to donate or feel you have something you can contribute, please contact any of the team members through the conservation page. Thanks for taking the time to view this video.